So here we have the cell of a photosynthetic bacterium. It's called the purple bacterium. And this wonderful presentation was done by Melik Sena, who sits there in the back, the tall fellow. And you see here a thin slice out of the bacterial cell that was done through ion beam milling. So you take an ion beam and you slice the cell, and the section here is about 200 angstrom wide, so it's very thin slice. And then you can look at this cell with excellent resolution in electron microscopy. And here you see sort of like I rotate it around in front of your eye that you see a little bit better the three-dimensional features. Through the ion beam milling you disrupt a lot of the structures of the cell, but you see also that much of it remained intact. And here's the nucleus of the cell, and so that's an empty space, but you see that much of the cell is filled with these circular things, and those are actually the chromatophores. And so if we now go and look at one more closely, then we can move there, and you see it's coming there into view. And so here is the chromatophore, and we began last class by summarizing the physical processes that we need to describe quantum biologically. And so I just want to show this now in images that I had done last time just through a list. And so this is this program VMD that you are also using for your case studies. You see I can rotate it around. It's not a movie. So up and down and left and right. So it's really, I do that now in real time. And you see here that it is made of the various proteins that contain chlorophylls. And the first one is actually the small green rings. And so we now zoom onto one, and you see what's inside there. So they contain 18 chlorophylls. And here you see one of them, you see how beautifully they are arranged. The greens here are the chlorophylls, and the orange is a carotenoid. That is actually half of the molecule that you encounter in vision. In vision, we have retinal, which is exactly twice as large as the carotenoid in terms of how many carbons are involved, how many electrons absorb the light. And so again, I rotate it in front of you so that you see a little bit better the three-dimensionality. And so then we have the next big elements in this chromatophore. Here's actually the entire element that is functionally important. So we have light absorption. That was our first process we needed to describe, and we did. Then we have the distribution of the excitation in terms of an exciton system. Those are the electron excitation that are distributed over those. Why did not? So here you see the chlorophyll inside there, and so you distribute the electronic excitation over those, and then you jump with your excitation from one of those excitonic excitations to the next one in another protein, in this protein. Then you jump over to the red one, and then to the blue one. And in the blue one, you have the reaction center that then charges the membrane that we described it last time. And then we charge electron on the quinone. The quinone moves to this BC1 complex. Electron is being shuttled back. and we turn then that potential that it was generated by electrons into potential generated by protons, and the proton gradient drive then ATP synthesis in this ATP synthase. And so here you see it sort of all, it's here beautiful, we know all the molecular components here. So it's really just like solid state physics, except the systems are more complex, and I would say also, sorry, that maybe my bias as a biological oriented physicist, more interesting. It's not that we don't know the system. So, you know, like people think that, oh, these biologists, they don't know what they're talking about. We know every atom. And I would say absolutely certain that we know our system, even though they are so complex, better than the solid state physicists know them. Because there's not that much interest in finding out structures of solids. So if you have a new solid, it takes years to get the structure, where there's such a huge interest in, from pharmacology, medicine, the life science in general, that these structures are being solved by the hundreds every day. And all these accelerators that you have today around, basically all are there for the purpose of solving biological structure. They're not there anymore for looking at some interesting physics processes. Very few of them, like at CERN, but most of the other accelerators are just there for biological research. So you really have here these systems where you know atom by atom what they are made of. And it's just absolutely amazing that we can do that, and you see that here very, very nicely, that we know these systems. And if I just go then quickly back into the cell, and you see there's a cell, and that's where we have, where we can see all these systems there. We know for the key systems of entire cells what they are made of, atom by atom, electron by electron. So that is really a huge change in the field of life science that comes about through physicists moving in and applying their technologies and their concepts. Okay, so let me then hide this away. And now I had this process. We want to understand how light is absorbed by the eye and gives rise to signal that goes to the brain. And so let's study that quickly a little bit. So we said that this is done through a molecule of retinal that is hit by light and goes from a conformation where it is bent to conformation where it is straight. And so here are the receptors. The short ones are the color receptors, the cones, the long ones looks like cucumbers. The long one, both of them look like cucumbers. Oh, this looks like a cob of corn, and this looks like a long cucumber, the rods. And so they contain those retinal molecules in the form of a protein called rhodopsin. Here you see this protein. And there's a retinal inside there. And it's surrounded by protein side groups, so-called amino acids. And their composition surrounding the retinal determines actually the color. So you always use the same molecule, retinal, and you just change the protein around it to move the color. So then I showed you the process that happens when light hits a molecule. The molecule changes its conformational state, and so we looked at it. And okay, so we're going to go into, and we have now this molecule, rhodopsin. This is the membrane, and so here's the eye, there's the retina, and here we have the rods and the cones. And the rods and the cones contain inside there these discs. The discs are made of membranes, and now you have in this membrane this rhodopsin. So it makes a lot of sense you want to fill this with as much rhodopsin as possible to really absorb every photon that comes by. So you have here this, you know, hundreds of disks in there. And one of the disks contains then two membranes, one on the top, one on the bottom, curved around, so they're connected with each other, look a little bit like pancakes. And in these layers here we have this rhodopsin. And now with rhodopsin we also know every atom. And so here we just do something that you are doing for the case study all the time. I just do it now here in front of you. So we have here the VMD program. You used it already for simple case studies. I make it a little bigger. And I also don't like the black color, so I quickly change the color here. So I have to go here, change the color to white. So here, so good. So now, why are they not changing it? Our background. And then white, okay, good. Oops, there. Okay, so now we have it in white. And then, okay, good. So now I can load the molecule. And so I go now here to file. You did that already several times yourself. And now I have to browse it. I have to find it on my computer. And so now let me see that I find it in fact. To find my Phys550 folder. So yes, Phys550. 2013, lecture 4, I remember, here it is. And so then I put it here in rhodopsin. And here I have the rhodopsin molecule here. And now I open the file and I load it. And here it is. So now I don't need this one anymore. And here it is. So if I make it a little bigger, then you see here the molecule. Now you don't see much, and the reason is that we have here everything. We have the water, we have the lipid bilayer. Inside there are divisible the protein. So to see it a little bit better inside, I first go here and show the different representation of the protein. So maybe I just make another one. And I make a protein. There's only the protein, and I choose a representation not in lines. They are hardly visible. Maybe I just do it as a quick surf as a surface. Where is it there? So, oops, there, and I color it in a little bit different way by res type. Okay, so there you see the retinal molecule inside there. Water on top. Membrane in the middle, water on the bottom. If I take the water away, then I go here and I put here just lipids. Then I don't see the. Did I write it right? Okay, good. So then you see the water is now gone. I might want to do the lipids a little bit differently, that we see it better. Maybe I do it, even though that's an expensive representation, as licorice. So we have all the chemical bonds of the lipids. So you see how they surround the protein. And if I take the lipids away, which I can do by just double clicking on this here, then I just see the protein. The protein retinal sits inside the membrane, and I can recognize that because I colored here the protein by the amino acid types. That's something you will be looking a little bit more in the future. And so they are positively charged, they're shown here in blue. They're negatively charged, they're shown in red. Then they are polar amino acids, and then they are hydrophobic amino acids that don't like water, they don't carry a charge. And they like the lipids. And you see that the protein has a lipid belt, has a hydrophobic belt, so it loves to sit there in the membrane. So if I add the lipids back, you see that exactly what they do. Maybe what I do is I make the lipids quickly transparent. So then you see a little bit better. Well, there's still the lipid there, but you see now better the protein. You see that just where the protein is very hydrophobic, that's where the lipids sit. Now, the action is of course inside the protein. So we just look at the protein, and I can make another representation of the protein. And so let me quickly do that. And now I want to see the protein more in its architecture. So I do that one by choosing what I call new cartoon. So yeah, incidentally, that's right. And now I color it by secondary structure. I don't explain much about this now. I do that later a little bit more. So I'm very brief here. Now I don't see anything because the surface of the protein is obstructing the view, so I can hit it away. And now you see, now this is the protein. I can add the surface back by putting it, but then making it transparent. And since I have an expensive computer, I can show off ski by making a display that shows the render mode at its most beautiful. So that is what poor students don't have, but the rich professor has his latest model, and you see that really we can see the surface, and we see also inside the protein at the same time. And now we don't see the retinal. Where's the retinal? So I want to see the retinal. What I do is I create another representation that just shows me now the retinal. And the retinal is, I know the name of it. I don't want to go into the detail now. But here we have it, and we put it in Van der Waals to show it better. Now I do it in that. Okay, good. I can do that, and I color it by atom type. And now I make the protein transparent too. Okay, so let me make it also transparent. And so now you see the retinal beautifully inside there. So if I take everything away, then we just see the retinal molecule inside the protein. And so this is a bent molecule that is being hit by light and straightens out and works against a protein that is surrounding it, and thereby change the shape of the protein. And then the protein begins to interact with other proteins to tell them about the fact that it's being that it has been hit by light and that is activated. And now the system works just like a photomultiplier that you see in a lab here in Loomis. The way a photomultiplier works is that you send electrons on a cathode and on an electrode, and then the photon liberates some electrons out of the electrode, and now you're putting voltage difference to second electron. The electrons come out of that electrode, move towards the second electrode where it is attracted by the voltage difference with a high speed. So now that maybe one or two electrons that have been liberated by the photon hit now with great speed into the next electrode, where they liberate now each of them 10 electrons. So now we have 20. Now there is now a third electrode where the same trick is being used. Again, you accelerate them, and now you have then 2,000, and then you have 20,000 when you go to the fourth, and thereby you can change a single electron or two electrons liberated by a photon into several 10,000 electrons that generate on your oscilloscope a real signal, and then they say, oh, I saw something. And that's exactly how it works here, except we don't have electrodes that amplify the signal. We have chemical reactions. So this molecule now activates 10 other proteins. Each of those activate 10 other proteins. So 1 goes to 10, 10 goes to 100, that goes to 10 other proteins, 1,000. And that final set of proteins now goes to the membrane of the neuron in which the whole system is and plugs holes where ions go through the neural cell wall. And when you change the conductivity, you change the voltage in the neuron, and that voltage difference is then measured, seen by other neurons, and sent to the brain. So you are amplifying. Then you use the most amplified protein to plug a hole in a neuron. The neuron then changes its potential. The potential inside out becomes much more negative. That potential difference is being seen by the brain and reported to the brain, and then the brain knows, oh, in this part of the eye there was an event, and there was apparently some light, and it puts that into the data bank, which is called the visual cortex. This is the area here, and it puts this then together into the picture of the world that is being interpreted through these photon events by the brain. Okay, so that is how it works. And again, what I want to tell you is here we see every atom. Let me quickly show you every atom that you realize. So I go here to that quick surf. I don't make it transparent. And now I instead of quick surf, I put Van der Waals. And instead of this one, I change by atom type, that we see every atom. White atoms are white, red atoms are oxygen, sorry, white atoms are hydrogen, red atoms are oxygens, light blue are carbon, and dark blue are nitrogen, and yellow are sulfur. And now I scale it up, and now I rotate it a little bit in front of your eye, and now you see we know every atom there, every atom. So that is what we know about the system. Yes, so if you want to know, ask a question, go to that structure and ask the question and answer it. So this is not fun and game. This is absolute, brutally well-characterized system. You don't find it better anywhere in science, particularly also not in other fields of physics. Okay, so this is now our roll-up scene, and you see how easy it is to look at it. You may think that, oh, this professor, he makes it look so effortlessly because he's so good. Ask my students. I'm the worst in the group, so anybody else can do this much better than I. So with VMD, you can really look at it yourself. You can enjoy it, you can explore it, and I'm not good at it at all. You can quickly become as good as I am by just using VMD for a week, and you can look at your own proteins and see that you understand them. Okay, so now let's continue a little bit more our journey into the vision. And so let me see where I was. So this was a process, and okay, that's what we all did already. We were here. So this is a process that takes place there, and now I show it to you for the nth time. And now here comes the physics. The physics is that that actually maybe I go back to this picture here. So here you see that you excite the molecule. We are now in the red state, and now the red state in the blue state, the red state in the green state interchange. This was the ground state of the electrons. We put in a photon, we go to the excited state. The excited state has different physical properties. That's why the molecule is now changing its conformation. Here it is totally, it's unhappy with the conformation, but it cannot do anything about it. When it's excited, the molecule can change the conformation. It does it. And so it does it, it changes over time. And then it comes to a point where the ground state and the excited state intersect. And now we continue on the ground state. This is now, this green becomes now the excited state. I might have better called this one red and this one green for the way I will describe this process. But we get here an intersection of two states, energy state 1 and energy state 2. And we want to ask ourselves, what is our chance that we are going along 2 or that we go along 1? So the probability is this one, let's say we call it P11, and this one we call P2, this one we call P22, this one we call P21, and then P22 plus P21 is 1, because we either go here or we go here. And no other possibility. And now we would like to calculate this probability, because we want to go here. We want to be sure that we go here, not that we go here and then go forth, back and forth and back. We want to go quickly down the ground state. And I told you that it's important that we are in the ground state, because this is a stable state. You cannot, there's no other state below it. It's infinitely stable. But we want to be on the right side of it. Here we have a protein that has different properties than on this side. And so this is what I show here when I show it now. So this picture here, we come from the top to the crossing point, and now we would like to go to this side where we have a new ground state. We don't want to go to the old ground state, because the old ground state, the molecule was not signaling. Retinal was bent. And we was not happy in the rhodopsin, but it didn't move the rhodopsin to become a signaling state rhodopsin. Here it does. And so we would like to know how is the chance that we go there. Okay, so that's what we have to describe now. And so let's do that then. So, so now I will turn this off for a while, and maybe somebody can turn on the light, and then we want to do a little physics here. So we are now in vision. The vision was covered by some pretty slides. But now we have to do a little conceptual work, and we want to understand the process of potential energy crossing. So this is the old state, and we are exciting the molecule with light. And here is a ground state. We call this one, I call it V1. Sorry, I call this a 1 state, but it's a ground state. Call this one the 2 state, just to have a more natural labeling for the mathematics I'm doing. But this is a ground state. The ground state here, in this up to here. Now excited by light, and now the molecule goes here. And now I don't want to go the molecule like this. I would like to go here. And this probability is called P22, and this one here is called P21. because here I have the state 1, and here I have the state 2. 2 becomes a new ground state, 1 becomes a new excited state. They cross. And this process we want to describe. So if we look at this one, then I draw it here again. And we have here the, I call this one the adiabatic picture. And why I give it this name, you will see in a moment. So this adiabatic picture, this is how we go. Now, if these states do not interact with each other, then that's the whole story. But these states interact with each other. Now, the interaction is small, but it becomes very relevant when these states become isoenergetic, because then the smallest perturbation can mix them. If you have a perturbation that is this big, you cannot mix it here because the other state is too far away. You cannot mix it, you cannot mix it. Now you can mix it, because now the states are within that window. Mix, 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 no mix, no mix, no mix. At this moment we mix it. So we have an interaction between state 1 and state 2 that we give the magnitude small v. Okay, and so now we can have actually two pictures. We can say, oh, this is one way of describing it. And then we have another way of describing it is by, we call this a diabatic picture. And in this picture, we look at the Hamiltonian V1, small v, V2, and diagonalize it. Because the stationary states are always corresponding to the diagonal form of the Hamiltonian. Here we want to ignore the kinetic energy of the system because what is really moving here are the nuclei of the system and not the electrons, and so we can ignore it for the moment. So this is what we have. Now we know what the energies are. The energies are E12 are equal to V1 plus V2 over 2 plus minus square root of V1 minus V2 over 2 squared plus V squared, small v squared. And so if I plot this one, then what I'm getting is, maybe I draw this one quickly. That was the adiabatic picture. And now the diabatic picture is this and this. So here, now I'm looking at the Eichstätt, I mix these two states, but here I have a pure one state, and here I have a pure one state because there is no mixing. Mixing is only here. Here I have a pure two state, and here I have a pure two state. But in the middle they mix, but there's a difference, a separation by 2V. And so they are separate. Now you ask yourself, and this is a good picture for stationary states. Stationary means slow. So if I go very slow, that's what diabatic means, do it slow. Then I should look at the stationary state description. And that would be the description. Then you would expect that if we are doing a slow one, I move actually like this, because this one is away. But if I move fast, so this would be slow, small velocity. And if I have a high velocity, I move through that really fast, then this one should be the right picture, because that's stationary, it takes time to become stationary state. I don't have time for it. I just go here, and this will be a high velocity. So now what is in between the situation? So the way you can describe it is that you are looking at the system and you define here the only thing that matters is the behavior near the crossing point. The other one is totally unimportant because there's no mixing. So here we assume a linear expansion. And so now in our picture we have now that we have two states, and we have one state is V1 tilde, or maybe I just write it like this. We have a V, and, Hamiltonian operator, which just gives energy, which is V1 plus, so I call it V0, 0, V0, and the V0 is just V1 plus V2 over 2. So this is just this constant value here. At this value. And now we have then plus. And now we have here a second one that is F times eta and minus F times eta. So I'm getting one positive and one negative linear term. If I make F negative, then you get plus F on the top. So it's okay with the choice of F sign. It's not really very important. And here I have the V. And now what I can choose, eta is if I call this one the coordinate x, then eta is x minus x0, x of t minus x0. So when I move through here, then this is my coordinate there. And now what I do is I make x of t equal to velocity times t. So I use this funny notation because I have, capital V1, V0, and then this small v. So I want to be sure that we don't get confused. What is velocity? What is interaction, and so on. So I write it like this. So no, this one is no a time-dependent perturbation. But it's the solution of this is surprisingly elegant or complicated, whatever you want to call it. I would call it elegant, and you might want to call it complicated. So it's not very trivial. You cannot just do simple perturbations, so you would get it all wrong. But you can do it, and we will provide you with a brief derivation of it. But you don't have to really read it, but the very famous derivation, the derivation is due to Landau Sena. And so you call this one then the Landau Sena transition. And now I want to give you the formula for it. What's the probability of P21 that we are going like this? And now I should remember it, but I don't. So we have to do this very carefully. You'll see from the formula that one has to be quite careful about it. And it is equal to 1 minus e to the minus, you would have never guessed it, 2 pi v squared, this is our interaction, divided by h bar, velocity, times f. So f is the difference of the slope here. So the difference of the slope is 2f, so half the difference of the slope. So if you look at it now, then you find that if the velocity becomes very large, then this one becomes very small, and then we have e to the 0, which is 1. We get 1 minus 1, 0. So if the velocity is very high, we go like this, what we expected. We go like this. If the velocity is very small, then we have here 1 over 0, means 1 e to the minus infinity, then this one is 0, then this is 1, and then we go like this. And we have the case in between. And how does the velocity relate to the other quantities? Here we see the other quantity that counts is the interaction energy. The interaction energy is very large, then this one is always very small, then we always stay on the top. They are too separate. They become so separate that we cannot really cross. If it is small, they're very thin, then we have a very nice time to get through there. And then you see also the F is important. If F is large, then this is 0, and then we also stay there. F is sort of like a little difficult to understand why it is. Basically, F determines how long are we in this region. And if F is very large, then the region where our V can couple the moment is very short. And so then you just cannot couple and you stay on the top. Whereas if F is small, then they are very flat, the two curves. Then you have a long time to couple, and then you have much better chance to go down. So you see that how this all depends. And so you see this is really the key point of controlling the retinal. You have to make sure that when it comes to this point, that it really goes through this point. Now this is the simplest description where we are not going to ask ourselves, what is the chance that we go here and not here? And so in order to get it more refined, you have to do even more work than the derivation that we give you. And we give you a reference where you can find it. And we are not going to do it, but it is discussed in the book chapter that I gave you last time, that I posted already on the website. If you're interested, the book chapter is called Quantum Biology of Retinal, and that's very nicely described. So if you're interested in this kind of stuff, go there and you'll find all the information. Otherwise, I give you here the key picture. Okay, so that you need to realize. Now, if I look at the retinal molecule, then this is how it looks like. This is how it looks like. I hope I didn't make too many things. I think I'm correct here. This is how it looks like. This is called 11 cis retinal, because you begin counting here, and then this one is number 6, 7, 8, 9, 10, 11, 12. Here you have the two hydrogens on the same side, whereas here, when you look at where the hydrogens, you find they're all on opposite sides. So here is actually, sorry, I made it wrong then. You have to then draw like this. So this is how it is, so I made a mistake, I realized it. So here, this is where the hydrogens are. So they're all on opposite sides, you call them trans. You go here, so you go here along the street, and you see one time you have a tree on the left, then on the right, and the left, and on the right, and then you have a tree on the left, and a tree on the left. That is where you have cis. And that makes a huge difference, chemically speaking and conformationally speaking, and so that's why you assume this kind of shape. And now you go to this shape. And here it is connected to the protein. So I don't draw here what is happening. It's actually a side group of the protein. Actually, it's a so-called lysine side group that holds it. And so the end of the lysine molecule, the nitrogen, becomes actually the first molecule, the first atom of the retinal. And so now we have this molecule. What we did is we rotated the molecule around this bond. And you find then, if you rotate around the bond, this goes to the other side. And then we have 3 to the left, 3 to the right, and then we continue. This one, since it rotates around, this one is not there. 3 to the left, 3 to the right. Now you see that if I draw here the hydrogens, then you find here trans, 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 everything is trans. You call this one all trans. But why don't you rotate around this bond or around this bond? Why do you rotate around this bond? That is what the system is designed into the system with our eye. And so that you are picking this particular bond for rotation. So you may think, oh, yeah, good, well, I studied it, and yeah, the professor told us last time, these molecules, they don't like each other, these atoms. Here's a CH3 group, here actually three hydrogens is the carbon, and here 3 hydrogens, and they fight with this hydrogen here. They're getting too close, they don't like each other. Yeah, that's one reason why they go around. But that actually, just to show you, life science is really a system that is really full of new discoveries and new development. Every day you open the journal or you read the New York Times, you find something. Here some really important discovery, just I think now 2 years old. You call it, what do you call it? I think you call it optoneurobiology. I think that's what it's called. So basically, people discovered an ion channel, like something like rhodopsin, except the retinal that is in this channel controls actually an ion channel. So when you shine light on it, that protein conducts suddenly ions. And now think of the potential. Many people didn't, but some people did. And so that they thought, oh my God, I put this protein into the brain in certain neurons, and then I shine light on the brain, and I can make the neurons change their potential, activate the brain, and make the mice twitch with the left hand, with the right paw. And exactly that's what they do. They put it into mice, they put it into many other species, and now they shine light here, they shine light here, they shine light there, and they can make the neurons change their electrical potentials and thereby change the activity and the working function of the brain. And so this is one of the hottest fields of neurobiology today. So I would say probably 1/3 of the field is really using this molecule. But in this case, you have a molecule where actually the protein is not very selective for the bond that's isomerizing. So there, to the surprise of the people, you can have all kinds of isomerization. We have in the eye only this isomerization. Now, people went to there's this famous guy the one who solved the human genome. There are two, but the guy did it by himself, sort of. What is his name? Greg Ventnor. So he's a very big, towering figure. in modern science. He's sort of like an entrepreneur scientist. And so he has a big institute where he was the first one to compete actually with NIH to sequence the first human genome. And he had a big ego, so that the genome that they were actually sequencing was, you may guess it, his own. And he makes a lot of money, and he has a big yacht, a sailing boat, sailing vessel. And okay, I just tell the story probably wrong, but I just for the heck of it, maybe he wanted to get a tax deduction for his yacht, and so he used it to go out on the ocean all around the world to do something sort of stupid, but with a very, very surprising result. He lowered a bucket into the sea, pulled it up, and then he analyzed on the boat what kind of bacteria, what kind of life forms were there in the sea. And so he did it all over the ocean, and they expected, they know what is in the ocean, they expected certain results. To their surprise, what they found was totally unexpected. They found bacteria that contain retinal that are by far the most abundant in the ocean than on Earth. So from one month to the next, when they reported, suddenly people realized that the most abundant bacterium on the world and the most abundant life form is one that is having retinal. Now what are they doing with retinal down in the ocean, looking around and seeing how pretty it is? No, they use it actually for energy purposes. They use the retinal to use the light energy that is being absorbed to actually pump a proton across the membrane of this bacteria, just like you pump protons with a photosynthetic system by first conducting electrons and then doing the protons. These molecules can conduct protons straight. And there you have a system that has this retinal straight, at a straight all-trans retinal, and when you excite it with light, then you are making from all-trans, you make a 13 cis form, and only 13 cis. So you rotate it, and then it pumps a proton through the protein, and then it thermally relaxes back to the straight form, and then you can absorb the next photon. It turns around. So you really play with it. In one case, only this one, from the 11 cis to all trans, one case from all trans to 13 cis, and in the optoneurobiology, you have one that does all the bonds that are available. And so you see there's a lot of things going on. So probably this is how this protein developed, how this retina developed. It was first used by bacteria to pump protons, but then life forms found better way of pumping protons more efficiently. But they remember this is a pretty good absorber. We don't want to get the energy out of it. We want to get the signal out of it. So when you have a sensor, it absorbs maybe also light, but you're not interested in the energy, you're only interested in the signal. So most higher life forms, they use it, but they are getting only a signal out of it. Whereas in photosynthesis, they are getting energy out of chlorophyll-based and carotenoid-based systems. So you see there's a lot of interest, and there is also important to understand all the potential you have. Rotate around this bond, rotate around this bond. And so here the question is, why does it rotate around this bond? Why doesn't it rotate back around 11 cis? And so you have then the following situation. You have a ground state, you have an excited state, and this is all trans, let's say. And now this bacterium goes here through a crossing to 13 cis. But why doesn't it go to an 11 cis? or a 9th or a 7th. That's actually what this opto-neurobiology system is doing. And so it goes here. And so this is where nature designed the protein to lead the system the right direction. Since this is a very fast process, so this one is actually for retina really slow, where this one here, this one goes to here. It goes in not quite 10 femtoseconds, but roughly 10 femtoseconds, so more closer to 10 than to 100. So it's very fast, so your protein has to be really good at it. It has to be absolutely designed for it. So how does this happen? And that you are very efficient in crossing here, that is the secret of vision. In this case, is the secret of vision. In this case, is the secret of by far the most abundant life form on Earth that gets the energy out of this retina. If I would have told you this like 5 years ago, and somebody across Green Street would have heard what this Schulten guy is saying in Loomis, they would have said, my God, these Loomis, these physicists, they know everything, they went totally bananas. And I would have agreed with them, but they all must have gotten totally drunk when I said it. But now, after they checked it out, it is really a very common form of getting energy out of it and playing with the system in very interesting physical ways. Okay, so then let me complete my vision here. You should be very impressed with bacteria. Probably many of you know this, but you know that you have more bacterial cells in your body than human cells. You know this, but that is so. They are all over the place, and we think they are our enemies, but you know we use them really. We live with them together, and they are amazing things that exist in biology that are just being discovered as we speak. So it's really a very young science. Okay, so let me now go. And so now I told you about signaling already last time. I don't want to repeat it. So basically we have here then absorption, and then we amplify this to make one event in one rhodopsin, to amplify it to change from one rhodopsin 10 proteins. They're called, the first protein that is changed is called G protein. And then we have 2 further steps so that we are amplifying one event, 1 protein changed, to 10 protein changed, to 100 protein change, to 1,000 protein change. You know, it's just not a factor of 10, it's a little bit more than 10. So we are getting at the end 10,000 proteins change. And they go then here to the membrane of this neuron. This is a neuron. And now the neuron constantly tries to keep a potential, and so it pumps protons, positive protons out to make it negative inside. And now when you suddenly block the potential, then you're changing the flow of the neurons. The neuron is still pumping, but now the backflow is altered, and then you are getting not the normal resting potential of the cell, but you're getting a much higher resting potential, and that potential moves towards the cell body here, and now if I go further down here, then the remaining proteins that are making contact here are sensing that change in the potential, and then they tell the brain. Now this is actually very, very interesting. This is another one of those stories in the life sciences. So all neurons, when they talk to each other through electrical signals, we will talk about that briefly. In October. They do that through through chemicals. So you think that they talk each other electrically, but no, this cell gets a change in its potential, it releases a chemical, and that chemical changes another cell that then says, oh, this cell must have been excited, I got all these chemical signals from the cell, and then it gets excited too. Now, a little complicated. Why do you think they do that? We have in our brain 10 to the I think 13 neurons. Quite many neurons. And if they would all talk to each other electrically, there's too much of a chance that there's crosstalk. So it's much better to have them speak English, German, French, and Spanish, and you have it. Because then you make sure that the Spanish guys talk to each other, the French guys talk to each other, and there is much less chaos. You can keep them apart much better. So really important, really good trick by nature to keep the communication pathways neat.
So they use different chemicals?
What?
They use different chemicals?
Yes, different chemicals. Yes, of course. Yes, okay. Yes, exactly. You call them neurotransmitters, and there are many types of neurotransmitters. But there's one exception, and that is in the eye. The eye is a very important sensory organ for all animals, really important. So we have the visual cortex that gets all the signals from the eye, and the visual cortex is about 10% of the brain. It's really a very, very active, very, very key area. So there's this important organ, and in this organ, you have actually electrical communication, because you know there is only one type of cell. You want to know if it got excited or not. You don't need the neurotransmitter bit, because there's only one issue here, light or not light. And so you have now electrical sensing. And so this is, so you may say, oh, but you know, this is a big deal, that usually they talk through chemicals. Here they talk through physics. And now you have here just proteins that can tell from this cell to the next cell that they got excited. They were just being discovered now. So the other day, I got a phone call from a guy who said, oh, Schulten, I have this really exciting protein, but I cannot tell you anything about it. It's too secret. Maybe I get the Nobel Prize. But can you work with me anyway? He didn't want to tell me, but he wanted me to work with it, because it's such a big deal. And they're just being discovered now. And so basically, people have now a very new class of proteins where one cell can actually directly communicate its electrical potential to the other cell, whereas in other ways it always is chemical. So really, and that just happened the other week. So this is really the nice thing about physical life sciences, that is really every day almost we are in the middle of new discoveries and things that change our picture. And okay, so here we have now the question: how can one molecule, retinal, absorb different colors? So here you see the colors that are being absorbed by the different color receptors. You should know that a human have 3 color receptors: blue, green, and red, and a black and white receptor that is for night vision. So we absorb at 4 wavelengths. You see that we have a little bit of a problem here. The blue is really in the blue range of the spectrum, but the red, red is 600, not even 600. Maximum is shorter than 600. So red is actually difficult to see for us. The real red end of the spectrum is 800, is here. So we really have a hard time seeing red, and but we do it. But we have to somehow, we can assign a color red, but it's a little difficult. Sort of, we don't have the sensitivity there, and so on. Okay, so how do we get the difference? And the difference comes about through surrounding environment of the retinal in the protein that shifts the spectrum around. So retinal is a good absorber of light, and it can have its spectrum easily shifted. I told you that already. And now you can imagine that things can go wrong. You know, like if you need a green receptor, you have to have a certain side group here that shifts it to the green. And if you are colorblind, then you have just a mutation here that has a wrong amino acid here, and then you cannot see green. Your spectrum is not shifted properly, and so that is in fact what you have. You know, here you see, for example, a famous test that you do with the eye doctor, and so this is what you would see if you're not colorblind, and if you're colorblind, this is what you see. And that's actually interesting because it explains a little bit why is it important not to be colorblind, or with other words, why is it important to see colors? Is it for entertainment value, that it's all this beautiful jewelry of the pretty women, or the nature, we can enjoy nature better? No, it is, you can study it with monkeys. Monkeys, you know, when they you know, they live in a habitat and they're constantly hunting for food. And one of the most important food sources are for them fruits. And they need to know if a fruit is ripe or not. And so they see there a red fruit up there. They say, oh, this is exactly the ripe, it's ripe, now I can eat it. And they climb up there and eat it. Whereas if they don't see the color, they climb up there in vain, you know, another one not really ripe. Maybe they even get indigestion from eating not ripe fruits. And so it is important for hunting for fruits that you see the color. Of course, you can also see maybe better your enemy. And so there are several reasons why you see colors, but there are many animals that have fewer color receptors than we have. There are some animals that cannot see color at all. So it's not absolutely necessary. And the 3 color receptor is unusual. We are very well endowed. Might be some insects have more. I don't want to lie now, but okay. Insects, by the way, have an ability not only to see the color, but also to see the light polarization. So they can see not only, you know, what color was absorbed, but also how was the light polarized. Do we have any idea why they want to do that?
It could help them know how close they are to water, because the light's polarized when it reflects off the water.
No, could be, but that's not the reason. The reason is that the light in the sky that is scattered down to us is actually polarized. And so if you see the sun, you don't need to know, you know, for roughly sun south is good. But if you have a very cloudy day and you want to know, the bees want to know where the beehive, they need to know direction. And so then you see that the light is actually polarized, and they can detect this polarization, and then they know where's south and where's north, because it shifts around with the sun. And that will be actually very likely your final exam, on your final exam, because the notes that we gave you on interaction of molecules with light contains, in the note that you got last time, also something about light scattering. And there's Rayleigh scattering, and that is answering the old question, why is the sky blue? And so that is explained by Rayleigh scattering. Look it up, Google it. Don't explain it now. But then you also, with Rayleigh scattering, you can figure out, and that will be your one exam question for you, what is the polarization pattern in the sky? And you'll find it is actually polarized, and insects use it. Okay, so we have this one. We have this one. Okay, good. Yeah, here this one I showed you already. This is that frog that sit in the water, and this is what it sees. And now it has in one part of the retina it sees the sky, and the other one the water, and the spectra are shifted, and that guy actually uses then what is called retinal 2, where you see that there is an additional double bond here. That means here we have a hydrogen, here we have a hydrogen, but if we draw it this way, then here we have two hydrogens, two hydrogens, two hydrogens, and here, however, we only have one hydrogen, one hydrogen. So we have, this one is sp2 hybridized, this one is sp3 hybridized, and so here the electrons go all the way from here to here. So if you count the electrons, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Whereas here we have only 12 electrons. So we have a longer molecule, and that absorbs more in the red. Okay, so then this is then the last slide for the vision. And so next time we will begin a new chapter, namely a very important chapter in biological physics, namely stochastic processes. So there's a lot of stuff going on in the cell. The molecules are very, very busy and working together, but the molecules don't have eyes, they don't have vision. They are blind. They're doing everything stochastically. So life forms became masters in working in the total dark, in working without, guided by directions, by just using stochastic processes, and still using them so well that things work, things find each other, things work actually very fast. And we will study over a period of 2 weeks how they actually manage to do that. Okay, thank you very much.